There's a molecule within your body that is essential to your development. Without it, you might have died, or have had abnormal mammary gland development or abnormal vascular development. The absence of this tiny molecule has huge effects. What could this ever so important molecule be? It's cadherin. So, today we're going to learn what cadherins are, how they work, and why they are so important. Before we dive any deeper into the exciting world of cadherins, we want to know what you remember about them. In class, we learned that cadherins are important in cell-cell adhesion. Pause the video and list a few key concepts surrounding the role they play in cell-cell adhesion and why this is important. Alright, well done. Let's move on. This big green Lego represents a cell. Cadherins are cell adhesion molecules, which means that they help attach two cells together. They are transmembrane glycoproteins, and the part that we are interested in is the extracellular repeat domains represented here by the red Legos. Cadherin is a calcium-dependent adhesion molecule. It only functions if there is calcium around to link the cadherin repeats together. If there isn't any calcium, they don't work. In the absence of calcium, the cadherin molecules are flimsy, and thus fail to adhere to each other. The presence of calcium stiffens the molecule, allowing the cadherins to link up and adhere to each other. So, when there is no calcium around, and the cadherins are flimsy, a protein-degrading enzyme called a protease swoops in to rapidly degrade the faulty cadherin. This big guy is the protease. He ensures that we only have proper functioning cadherins around. Without cadherins, cells won't adhere together. Cadherins allow cells to aggregate, thus allowing them to migrate together as a group. Now, why did we use Legos to represent cadherins? Legos attach to each other just like cadherins do. They allow us to show the qualitative and quantitative differences that cadherins possess. Let's talk about how quantitative differences in cadherins affect their function. Here's a group of cells. Some have 4 cadherin, whereas others have only 2. The cells containing more cadherin will aggregate more tightly together than the cells with less cadherin. This is a way for cells to sort. In a developing embryo, cells with quantitative differences in cadherin sort into distinct layers. Ectoderm on the outside, followed by mesoderm and endoderm in the center. Now let's talk about how qualitative differences in cadherin affect their function. N-cadherin and E-cadherin are the types of cadherins we focused on in class. However, there are multiple types of cadherins, including VE and P cadherins, along with many more. These different types are found in different tissues in the developing embryo. In most cases, cadherins bind preferentially to the same type of cadherins. This is called homotypic binding. Typically, cadherins do not bind with other types of cadherins. This is called heterotypic binding. However, this does occur in few situations. There are 3 types of cadherin interactions. When cadherins on the same cell adhere to each other, we call it a cis interaction. When cadherins from different cells bind to each other, it is a trans interaction. When those cadherins are the same type, it is a trans homophilic interaction. And when cadherins on different cells of different types bind together, it is a trans heterotypic interaction. Now, what do cadherins have to do with embryogenesis? First, they allow for cell sorting in the developing embryo. Second, they allow for collective migration of cell groups. And lastly, they allow for boundary formation in groups of cells. When a distinct separation occurs in a cell group to create 2 separate groups, it is called boundary formation. Cells in the group are held together by cadherin molecules shown in blue. The green represents actin filaments, which are cytoskeletal structures that help support the cell structure. When a boundary first forms, cadherin molecules move away from the boundary. Then, the actin filaments move in and replace them. The reduction in cadherin allows the cells to detach along the boundary. The actin prevents the cadherins from moving back in, and the 2 cell groups are now separate. So, let's review what we just learned about cadherin. How does a lack of cadherin negatively impact embryo formation and lead to death, abnormal mammary gland development, abnormal vascular development, and diseases like pemphigus vulgaris? Now, pemphigus vulgaris is a rare and severe autoimmune disorder that causes blistering on the skin and on the lining of the mouth caused by antibodies being directed at cadherins, reducing their function. So, the lack of cadherin would lead to issues with cell-cell adhesion, collective cell migration, and cell sorting. which are all contributing factors to these conditions. Have you been paying attention? Let's test your knowledge with a question. Why did we use Legos to represent cadherin? Talk amongst yourselves and then share your answers with the class. Thank you for watching. Hopefully we have increased your appreciation for these very small but mighty molecules.